Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome back to the Genetics Podcast, everyone. I'm excited to be here with Dr. Jim Wilson, who's a professor of human genetics at the University of Edinb, and he's known for his work on the genetics of isolated populations. He's led the Viking Genes project, which explores the genetic history of Scottish Islanders. And he's also, this is very much not his first rodeo on public engagement and talking about science. He's done more TV and radio than we can count, actually. So I'm excited to a really great and engaging episode, no pressure. But I would love to actually start with an interesting fact that you shared. So I think you've been to dozens of different islands studying isolated populations, 25 plus as just part of the Viking Genes project alone. I wonder if we could start there. You've been to fascinating places. You've studied these isolated populations. What's been the most interesting or impactful work that's come out of this for you? Because you've published dozens, now probably hundreds of papers working in these communities. But I'm interested in what's been the most interesting or impactful to you so far.
Yeah, well, I grew up on an island in the north of Scotland, and my family have been there for hundreds and hundreds of years. So I've always been interested in islands. And I guess that underlying sort of deep interest is what drove me to start setting up these studies. So I came back up to Edinburgh in 2003, and that was really the beginning of what's now called the Viking Genes program. And we studied all sorts of different things. My initial interests were in population genetics and sort of in ancestry genetics and trying to work out where people came from. And that's very important to the people in the north of Scotland because they all believe that they're Vikings. And luckily for me, there was a strong signal of Scandinavian ancestry in their genomes. And then I sort of took a turn towards a more medical direction and really, I guess, became a quantitative geneticist in the era of GWAS. And we had a particular speciality in homozygosity, which is related to inbreeding, which again may be explained by some of my family coming from very, very small islands. My father actually comes from the most isolated inhabited island in Northwest Europe, Fair Isle, where the jumpers come from. And yeah, his genome's really quite interesting. He descends about 20 times from this one Wilson ancestor. So we were quite interested in this. And that was the most impactful work I'd been involved in until recently, because we had a Nature paper where we'd assessed whether inbreeding and homozygosity influenced a lot of the traits people had claimed that they did. And in fact, it turns out it's not as bad for you as people might think, although it does affect your height and indeed cognitive abilities, as well as a few other traits. But more recently, I think I've really become a clinical geneticist in some ways.
Yeah.
Because I've been interested in these so-called actionable variants. So in these pathogenic rare variants that carry in part a very large risk of disease, like, for instance, breast cancer, inherited high cholesterol, cardiomyopathies, and these things. And it's this work that we're really presently engaged in that I think will have the biggest impact. We keep finding more and more of these very strong founder effects, whereby certain such variants like BRCA1 or BRCA2 are really, really common in some of these islands. There's one island in Orkney where 1 in 20 people carry a pathogenic BRCA1 variant.
Wow.
I mean, this is crazy, right?
Yeah.
And it's just because of genetic drift over hundreds and hundreds of years and a lack of immigrants. And so the variant comes down, so it's probably a mutation happened in, 1650, 1700, and it's just been coming down the generations ever since, just like what happened in the Ashkenazi Jews. So it's this area that I'm really keen on at the minute, and I think will have the biggest impact because we're basically trying to put an evidence base together to try and change policy so that these variants can all be screened for, not just in people who volunteered to be in a genetic research study, but the population at large. I think they should all be screened.
Yeah, absolutely. I'm really interested to dive into that. The Orkney Islands themselves, there are hundreds, right? And but there are about 100 that are inhabited. Is that
There's about 100 islands, and I think only
yeah?
about 17 are inhabited, actually.
Oh, okay. I must be. I'm probably including some of the other
Or the other ones as well.
archipelagos
Yeah.
around Scotland. Before you started this work, it was probably already observed that, like with the BRCA1, for example, was it well understood that breast cancer was much more frequent on the islands and the genetics came in to help understand why? Or is it, so small and understudied that actually no one had really kind of done the work to understand what's going on in terms of these, founder effects? Because we've spoken before on this podcast about Finnish heritage diseases and other isolated populations where you have, and it starts with an observation of, hey, this is much more frequent in this country than some other places or this island. But I'm curious to hear the kind of clinical, genetic, and clinical scientific history of the islands and how that's played out.
Yeah. So most of the founder effects that we discover have already come to the attention of clinical colleagues who work for the National Health
Yeah.
Service, but they're always really quite shocked quite how common they are. Because
Right.
when we compare the families that we know about to the families that they know about, they're always missing half of them.
Right.
So we always roughly half, and I understand that's what happens in the Jewish screening that goes on in England now as well. So basically, doing the population-wide screening, you double up the numbers that were known, and also we are finding things that they simply didn't know about. Of course, for most of these diseases, so like BRCA1 and BRCA2 only explain a small proportion of all breast cancer. So it can sort of be hidden in
Right.
amongst the cancers that are just appearing by the by
Mm,
as well. So the really most common ones, they came to their attention in the last 20 years as well as genetic testing came into clinical practice in the United Kingdom. But the ones that are a bit more borderline, they weren't aware of, and we're finding them fresh. And in every case, we're finding many, many more people who carry them and really shocking frequencies. And this is important not just to people who live on these islands, because there's a huge diaspora of people. They leave to go to university, they leave for work. And
yeah.
so all across Scotland, the towns and cities of Scotland and indeed England and indeed in Canada, particularly, and in New Zealand, there are lots of people with Scottish Island heritage. So they bring their genes with them, and we start finding people carrying these pathogenic variants, but living in New Zealand, for instance.
Right.
So it's starting to spread across the world.
What's the population of the islands roughly that you study? I have no clue.
So there's about 23,000 in Shetland, maybe 21,000 in Orkney, and 30,000 in the Outer Hebrides, and smaller numbers on the Inner Hebrides. So what's that's like? Yeah, coming on for above 50,000. But I think there'll be, for all the people who live in the islands, there's as many who like myself, come to
Yes,
the
moved off.
islands but no longer live there. So the total population is a bit hard to estimate because we don't have a good measure of all those who left.
Yeah. So I'd love to hear a little bit about the work you're doing on screening in this population, return of results, because it's a big enough population that you have there's a lot to work with from a sample size perspective. I think about Iceland, for example, where they've done amazing genetics work in a population of 300,000 people, and you almost have a similar opportunity here, right, to understand the really kind of from an evolutionary perspective, really different population that can teach us a lot about fundamental biology, but also really help the people like your family on these islands that deserve access to screening that picks up. I think in the notes that I had from ahead of time, you mentioned that there's a cardiomyopathy that's 4,700 times more common in Shetland. That's not a typo. That's 4,000 times more common. That blew my mind. I'd love to just talk a little bit about what the screening is today, what you're working to get it towards, and what we can put in place there.
Absolutely. So I think things turned. So I've been collecting samples and running clinics in these populations for years. But about 5 years ago now, we entered into a collaboration with the Regeneron Genetics Center, whereby we were able to get everyone exome sequenced. And that just opened up so
Yeah.
much more than what compared to what we'd had before, which was chip genotyping, because we could see all the rare variants. And it's actually that BRCA1 variant from Orkney that really jumped out because we had 20 carriers in our cohort. And we're looking at this going, what is going on here?
Wow.
And that's when I began to sort of turn around the research I do and really point and do more of this sort of thing. So we then decided to do a more proper survey, and we used the American College of Medical Genetics list of genes for secondary findings. And when we have done this, we've actually found 10 founder effects now. So there's a BRCA1, the BRCA2. There's this Titan variant that you're referring to that's over 4,000 times more common in Shetland, and a good number of others. There are different ones in the Western Isles. So each island group has its own sort of founder effects, which I guess we would expect, but they seem to be particularly true in Shetland. Shetland is the furthest out into the ocean. It had a smaller population in the past, and I think it's had very, very few immigrants, really. So we see really profound founder effects there, more of them and stronger ones. So we saw that for these actionable variants, these variants that increase your own risk of disease and about which you can do something about. This may just be something easy. So hemochromatosis is the most common genetic disease. It's a disorder of iron metabolism that causes the body to load up iron, which is eventually toxic to the liver, particularly the joints, and eventually the brain. And the treatment for hemochromatosis is simply to give blood. Women don't become ill until after the menopause, usually, because they're losing blood through their monthly cycle, but men become ill earlier. And the treatment is really simple, and it's sort of like a virtuous cycle, because the donation of blood helps other people to become well. So some of it's easy stuff like that, obviously, for BRCA2, which can impart very high risks of breast and ovarian cancer for women, but also male breast cancer and prostate cancer in men. There may need to be more drastic intervention. People are offered mastectomies and oophorectomies if they want to when they're finished having their family. But in every case, there's something that we can do. They're called actionable because we can take action. Yeah. So we found the founder effects there, but we've also been looking at a different kind of variant that cause childhood disorders. So recessive variants. I'm actually a carrier of the famous Delta F508 mutation in the cystic fibrosis gene. Very common in Scotland, but of course I'm completely healthy. It doesn't affect me. But, I would need to be careful when I wanted to have family with someone that they weren't also a carrier. So we looked at some of these variants and we found numerous founder effects again in Shetland. Some of them really disturbing. There's a disease called Batten disease, and this is a rare disorder. The baby's born completely normal, but it's essentially a neurodegeneration. So it tends to go floppy, and then it's like really early onset neurodegeneration, and it's fatal. The child dies before, when it's a teenager. Really horrific disease. 1 in 40 Shetlanders is a carrier. So 1 in 40 Shetland girls, 1 in 40 Shetland boys. There's a non-trivial chance they get together and bring more Batten babies into the world. So as I realized all these things are at such increased frequency, initially I thought that people in Shetland should be screened for BRCA2, but now I'm starting to believe that they ought to be screened for a panel of different variants, both actionable and variants that aren't going to affect your health, but will influence your children's health all at once. It's an effort to get people into screening. You've got to raise awareness in the community and raise the funds for this, get the saliva. You might as well just have a one-stop shop and do it all in a oneer. So actually, I'm trying now to publish a good evidence base for what this screen would be like. At the moment, there's no screening like this available to these islanders. And it's actually an inequitable situation because the Jewish community, just as of last year, have a scheme called JBRCA for Jewish BRCA, whereby anyone with one or more Jewish grandparents who is cared for by NHS England can have a BRCA test, which is absolutely right and should be the case because they have 3 founder variants that altogether reach a frequency of about 1 in 40. And the critical thing here is that they don't need a family history of cancer. You can be tested. It's a population-wide test. You can be tested in the absence of that. So we need the exact same thing for the islands of Scotland and indeed the Jewish community in Scotland who can't at the moment take advantage of this. So it's giving me good arguments to have with politicians and with the NHS because JBRCA is up and running. And I'm a great supporter of JBRCA, but we need something like that, I think, in the Scottish Islands as well, and indeed for their descendants who've moved to Edinburgh, Glasgow, and all over.
Yeah, and I think there's similar programs around Tay-Sachs disease, right? Which to me feels like a good analogy to the Batten disease example that you had. So at first it made me think about newborn screening, but then it made me think even a step further ahead. I think in some Jewish communities, there's, premarital, genetic counseling effectively to figure out if you can understand if this is going to be a risk, plan around it through IVF with PGD. How much are you thinking about each of those different buckets right now?
That's absolutely right. The Jewish community are at the forefront of this, both for the BRCA, but also for these recessive variants. In the UK, there's a program called Genetics, and they offer screening in schools to senior pupils and also at universities and in Jewish community groups for 47 recessive diseases that have become more common through genetic drift in the
Wow.
Ashkenazi and in other Jewish groups. And I think that's the prototype that we'd like to aim for. I mean, I'm setting the bar high here because that's not something that really happens in Scotland anywhere at all. But why shouldn't it? I believe everyone will be in 50 years' time. Everyone will be screened. It'll be absolutely normal part of healthcare. But why wait? And if we're going to start this, you need to direct resources to the places that need it the most for a given cost of DNA sampling and genotyping people. You're going to get more bang for your buck if you start where the risk is higher, where the frequencies are higher, wherever that might be. And it's commonly in these sort of isolated communities. So, yeah, I'm very much looking to follow the path that's been blazed by various different programs in and around London in the UK. But of course, in the States and in Finland and in other places, a lot of this is much more routine. So I'm in a bit of a fight because there's not a lot of money going around at the minute in this sort of climate. But I think at the end of the day, I'm trying to persuade people that we will save money. Obviously, if you catch these diseases early and you can intervene, you're going to save the money of treatment later. And if you can prevent unnecessary birth of babies who are going to die, I think most people would be pro that. And of course, you're bringing, for the majority of people who were to take this test, they wouldn't carry an actionable variant and they wouldn't carry a recessive variant. And so they then be able to relax. And
it's great news too. Exactly. Yeah.
Great news as well. So this is where we're at. It's not easy to get these things funded, but I'm presently trying to raise funds locally in Shetland in particular. And I was just at Parliament earlier this week arguing with people there and trying to, the job is first to raise awareness. We need to make the population aware of this so that they can demand from their political representatives that's what they want. So I've probably got a long road ahead, but I think this is really important. I've become quite an activist actually about these sorts of things.
Yeah, I'm sure there are supporters and then there are people who it's hard to not support this in some way, but probably the easiest way to not support it is to just delay and not really get behind making it happen. Is it mainly a resource thing where everybody says, yeah, we love the idea, but and years go by and things don't happen? Or is there anyone who doesn't, who can't get behind something like this?
Yeah, so I think there's there's definitely a resource issue for sure. And not just in terms of doing the testing, because then of course you're loading up the downstream services, whether that be mammography or echocardiography, or usually some various forms of scans are required. And these services themselves, have financial constraints. But I think there's I dare to say, old-fashioned view in clinical genetics that's very much focused on not worrying people. People will say to me, but Jim, you're sentencing these people to a lifetime of worry. And I'm like, yeah, but they need to know. If I was a carrier of a hemochromatosis pathogenic variant, I would want to know. It means that I would have, eightfold chance of liver cancer. These are really important things. And what is the life sentence? I just go and get my iron checked once a year.
I give blood. Yeah.
It doesn't seem too bad. And so, yeah, I don't know. It's sort of like I'm wading into this area where some people are rather entrenched in their ideas. And of course, if this were to be rolled out more broadly, it would have considerable resource implications. But that's maybe another reason why starting in a place like Shetland, that's circumscribed, it's an island group, there's only 23,000 or so people there, get that could act actually as a pilot for, bigger scale operations nationally or regionally later. So that's another argument that I'm trying to use.
Yeah, and I am disclosing here that I'm using ChatGPT for this, so who knows if it's totally correct, but it feels correct. There's about 200 births a year in Shetland Islands, and that's not a lot of births to screen to, at the 1 in 40 carriers. You maybe get a Batten disease every 8 years on average, right? I shouldn't do public math, but it feels like avoiding that is worth screening a couple hundred people a year the cost for that is minimal, right? And even if you just focused on that one disease in a newborn screening context, I can't imagine the numbers not making sense.
Yeah, I mean, public maths is never wise, but I think your figures sound
Yeah.
roughly right to me. And what's important is that it's a drop in the ocean, right? The cost of
Yes.
this is a drop in the ocean compared to the expenditure across the whole of the National Health Service or even the Scottish part of it. And and the gains are for avoiding batten babies, but there's a number of other, we had found at least 8 conditions that also had drifted up, not quite as to such a high frequency, but obviously when you add them together, that's more that we would hope to avoid.
Yeah. I'd be interested as well to get your take on more broadly your engagement with not just Westminster, but also the kind of the British royalty and apparatus more broadly. You're quite famous for some work you did back in the day to map, I think it was Prince William's mitochondrial DNA. I'd love to just hear a little bit about the backstory there, because this isn't your first foray into into working with the powers that be in Westminster to try to do some interesting science.
Yeah, well, I'm interested in all sorts of things. And as I said, I started off as a population geneticist, and I'd always heard a story of Princess Diana having an ancestor, a woman called Eliza Havork, who was actually the maid, I think, to her ancestor who lived out in India, in Gujarat. And I thought this was a kind of unusual story and maybe not a very well known one. So I decided to track down a couple of cousins of Princess Diana who were related in the maternal line. So, of course, mitochondrial DNA comes from the mother's 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 mother, like this. So we could just track down to get descendants who would carry this mitochondrial DNA. And they knew that they were distant relatives of Prince William.
Yeah.
And they were quite interested. They didn't know the story about India. So we got it sequenced. And lo and behold, it was actually a South Asian, really quite rare mtDNA type, proving that Eliza, who actually there's evidence that she could write in Armenian, and she clearly had an Armenian father, Kevork, I think, I'm probably pronouncing it wrong, but I think it's George in Armenian, but it looks like she had some South Asian ancestry as well, I mean, for certain. And we actually saw in the autosomal genomes of these people, they were about 1% South Asian. They had a few little blocks of South Asian ancestry. So we'd predict that Princess Diana and therefore Prince William would also have these few little bits of autosomal ancestry. And so it was just it was essentially hobby genetics, something of a side interest for me, but it went completely wild.
Yes.
And I was on TV and on the front page of the Times, but I think it's only right and great that essentially he's in some tiny little bit of a way mixed race. And that's surely a good thing.
I've always found these and this is like your kind of population genomics background and now morphing into a clinical geneticist. I think until you've spent a lot of time in population genomics, you don't quite realize the implications of generations of trees going back and actually how many shared ancestors we have with one another, right? We have this concept of I'm from the United States, or you're from, the Scottish Islands. I'm from Britain, somebody's from Britain, but actually you don't have to go very many generations back for almost everybody to find a bit of everything in our tree. Maybe can you talk about that phenomenon a little bit? Because it's hard to wrap your head around without like sitting down and doing the math or looking at how the trees kind of braid together when you go back in time.
Yep, absolutely. This effects are actually magnified in isolated populations. So in Fair Isle, where my dad comes from, everyone on the island shares an ancestor who was born in 1775.
Wow.
So it's really not long ago. Then you go to one of the islands in Orkney. I think you'd have to go back to around about 1650 to get that effect. And in the whole of Orkney, you're getting this effect around about 1550. And then you get out to the country scale. And it comes down to the fact that we all, the number of ancestors doubles every generation. So you've got 2 parents, 4 grandparents, 8, 16, 32. And you don't have to go very far back in time before you've got 1 million ancestors. Yet, if you look at the population size of Britain or wherever your ancestors come from, there'll come a time where there weren't 1 million people there. And so it's called the ancestor paradox. And it's solved because, of course, lots of those people are actually the same individuals. And everyone is therefore related. Everyone is therefore inbred because your mother and your father are also related. And it's a bit mind-blowing, actually. And of course, people don't have pedigrees generally that go all the way back 1,000 years, but the royalty do. And when we've looked into them,
Yeah.
there's these projects on the internet now trying to trace, random celebrities back, and they all descend from Charlemagne, or they all descend from, King Alfred the Great of England, because probably we all literally do as we go back in time in order to fill in all our number of ancestors. And we'll descend from a whole load of people that we simply don't know the names of as well. But because the royal pedigrees were so well recorded, that's the easiest one to link into in this way. So yeah, I love when there's this connection between population genetics and the clinical, either in terms of the shared ancestry or in terms of the more defined ancestry, because these sort of ancestral groups we found in Scotland that we thought were just of general historical interest, we're now finding that this is exactly mapping on to where we find these founder effects. So
Right.
this group here, that's got the BRCA1, this group here, that's got the BRCA2, this guy's here have familial hypercholesterolemia. And so it actually is really important for your health today, and not just of historic interest.
Yeah, you released a paper. It's on medRxiv. We'll put the link in the show notes. But before we kind of move on from this topic, it would be great to just summarize that work has obviously come to an end of this particular chapter. You've returned results, I think, to more than 4,000 people. You found, I think, 7 genes that have substantially increased frequency. Maybe you could talk a little bit about what you kind of put out to the public in that paper, and then also what's next after this.
Yeah, for sure. So the paper's under review at the minute, but it's on medRxiv. We're the second cohort in the UK to be given ethical permission and to receive consent to return these medically actionable findings to individuals. So we tested, as you say, 4,000 people, and these cohorts were recruited between 8 and 18 years ago. So we weren't able, and we didn't take consent at that baseline for the return of results. That wasn't a thing back then. So we had to go back to them to consent. So of course, some people are lost to follow-up. Some people have died in the meantime. So the figures weren't as high as we might have liked, but nevertheless, I had 64 letters to send to people who had actionable genotypes. And we don't have a full follow-up of what happened, but we know of more than a few cancers that have been discovered. So people who had a letter from me, for instance, telling them they have a BRCA2 pathogenic variant, they go into their doctor and they get a mammogram and they find cancer right there and then. A man also discovered he had prostate cancer after receiving my letter. Various people have found, after having an echocardiogram, that they have abnormalities and will need surgery and medication. So it's really having real impact, immediate translational impact in the here and now. And so the paper describes the whole process. We're hoping it can be an exemplar particularly for British cohorts, because we're following British research ethics and the legalities of it all. The paper also describes these founder effects, and I think it's hopefully a strong plank in the evidence base for screening, because the screening is going to be very costly. If you just screened the 10 founder variants plus the hemochromatosis one that's common all over, you'd find the great majority of the people. So it would be cheaper than having to do exome sequencing on everybody. This sort of simplification of the gene pool that happens in an isolate is a gift. When it comes to screening, people used to think that the simplification would help in finding sort of the underpinnings of disease. And while that might be the case, it's really only in Finland and in Iceland that they've got big enough data sets and enough money to really chase that down. But it definitely is helpful if you want to go screening the people, because you simply have fewer things to screen for.
Right.
The risk has become concentrated, if you like, in this relatively small number of variants. And if you check all those variants, you haven't got the risk down to 0, but you've covered the great majority of the risk. In Orkney and Shetland, 93% of the risk of breast cancer coming from BRCA variants comes from 2. So 2 variants covers 93% of the risk. In UK Biobank, it's over 350 variants that you'd have to cover to get that much of the inherited risk covered. So it's not just small effects; these are huge effects. Yeah. And I hope that we can take advantage of that, the sort of natural experiment that nature has given us, and we should do what we can to protect these people.
Yeah, the thing that this also got me thinking about is how many of these other natural experiments are there around the world? Do you have a sense of how many other isolated populations there are? I'm familiar with a lot of them: Iceland, Finland, Quebec. There's Greek islands that have a similar effect, but I mean, there's got to be hundreds of these different communities, right? What do we know about the sheer number of these different places on Earth?
Yeah, I think there's really, really a lot, much more than people perhaps imagine. There are many different kinds of Jewish communities that are all, Yemenite Jews, Georgian Jews, and not just the Ashkenazi Jews. And India, the subcontinent, is a particularly structured place. And David Reich and others have done work showing that there are numerous populations in India, numbering sometimes, because the scale there is crazy, right? Sometimes numbering in the millions of
Yeah,
people who are more at least as drifted as Finland is. Just to
right.
take Finland as a yardstick here. And so I think it's not very well known. If you think about the Pacific nations, there's lots of founder effects obviously going on the Pacific Islands. And I really think it's way, way more than people think. You're going to laugh at me, but sometimes when I go on holiday, I like to go to a graveyard of the place I'm
Yeah.
visiting just to get the feel for the place. And everywhere we go, like in Italian towns, they all have the same surname, right? And so they're probably an isolate that's never been studied. Then go to a Spanish town or the Canary Islands, and it's the same. And obviously, they're different surnames in different places.
Yeah.
But this phenomenon is much more widespread than people imagine. Obviously, most people nowadays live in cities, and they're mixing and assimilating, and the gene pool are being mixed up. But nevertheless, a substantial minority of people are still living in traditional societies or on islands or in mountain valleys in the Alps or other mountain ranges or so on and so forth. So I think and you see these BRCA founder effects all over the place. So I think this is perhaps underappreciated by, urban society at large.
Yeah, yeah. I don't visit the graveyards when I go on holiday. I avoid them, but maybe I'm going to change that up. It's an interesting observation. I one thing I noticed in the paper as well, which is just kind of interesting to me, is that you had definitely more than half of people respond and want to get the letter back and consent, even though it had been years, and only a very small number of people actually responded and said, no, I don't want the results. That's interesting to me, that also lends evidence to, look, we should consent for this up front. We should proactively offer it. I think the newborn screening programs, Guardian in the US, put out some interesting data that the vast majority of parents want to get this information, assuming it's actionable and all those kinds of things. So I think people are, to the point you made earlier, asking for this as well, pretty clearly.
So we have an online cohort we recruited in slightly more recent times, and we did consent them at the top of it. So there's 6,000 people recruited. 98% said they wanted to have the results returned, and we're doing that, like that's ongoing in these months and the early part of next year. And 2% said no, but they exercised the right not to know, but they can change their minds. They can come back and change their minds if they want. So it's really an overwhelming, actually, desire to know this information among the people who volunteered to be in the research study. So I was very pleased with that. You want to be doing something that the people want. We have a PPI committee, so a public participant interaction and engagement committee, and they also strongly supported this kind of work. We surveyed a couple of thousand people, and they could rank the things that they wanted us to work on, and this was ranked very, very highly, understanding the distinct nature of their gene pool and returning actionable findings.
Yeah. One more interesting story I wanted to cover. You won an Irish BAFTA, an IFTA, for some of the work that you've done working with an Irish traveler community. Could you tell us about that? I was just interested to learn more.
Yeah, sure. So I've been involved in various strands of TV work over the years. I don't know how it comes to pass, but it does. But this one I really like because the Irish travellers are an isolated population. And we, off the back of it, actually ended up getting a paper published where we demonstrated that they were genetically quite distinct, forming their own little island on a UMAP or a principal components analysis. And indeed, it turns out they have elevated frequencies of all sorts of diseases. So they're quite an impoverished community, the Irish travellers, and there's a lot of racism, unfortunately, in Ireland. But eventually, in the Irish Parliament, in the Dáil, they were recognised as an ethnic group. And that's not because of our work, but I like to think that we contributed just a tiny little bit to that recognition. It was really interesting to make the documentaries and go out and meet all these Irish travellers, because they've got a really lively sort of folk heritage and oral history with music and song and stories. And I love going out and meeting people, especially people from smaller communities. And, it reminded me of islanders, actually, because they're a genetic island, but not a physical island.
Yeah,
but,
it's a cultural island. Yeah.
Island and a strong sense of community. And I was privileged, actually, to meet. I meet a lot of these people during the making of those two documentaries all that time ago. But then I was very pleased that we got a scientific peer-reviewed paper out of it as well to really put that on a firm footing that they are distinct. We're presently studying the Scottish travellers as well, with a view to understanding whether they are distinct genetically or not, but that work's ongoing.
Yeah, interesting. This is fascinating. You have so many stories. Any other interesting stories that you think people would be interested to hear before we close out?
Oh, I don't know. I hardly know where to start. Yeah, I worked in many places over the years. Yeah, I'm not sure which one I would highlight. We've covered quite a lot already.
Yeah, we have. Your work is fascinating. I think it's cool as well how you've managed to it takes you to different places in the world as well, right? You can really you can see the world and experience all these different cultures while also understanding the role that genetics plays and how it might contribute to improved health. I really like this insight about the efficiency of screening because these communities are often left behind by the fact that they're smaller, they're geographically marginalized in many ways. They may be part of smaller countries in some cases that don't have the resources of the US or one of these bigger countries. But if you can turn that weakness into an advantage to say, actually, you don't have to do a whole long read genome to figure this out. There are 10 variants that can get us 90% of the way. If we just figure those out, we're going to do a lot of good. And that's something I hadn't really appreciated before.
Yeah, I mean, it's great, isn't it? Because we're turning it on its head. So they are commonly marginalized, whether geographically or to do with money or whatnot. And this would be a way to balance things up,
Yeah.
And bring them to the forefront of genomic medicine and preventative medicine, which is where we want to be. We don't want to be treating people when they're ill. We want to be finding them ahead of getting ill.
Yeah, and I think drug development companies should be really interested in studying these populations. I know they are. You've got a partnership that you mentioned with Regeneron, but if you were to sequence 1 million people from isolated communities around the world versus 1 million people from a broad population, you kind of want both. But we've already got the first one in the UK Biobank and Our Future Health and all of us. I think an isolated community million genomes program would teach us so much about so many diseases, right? Because you only need 1,000 people in that group of islands like this to discover that whopper variant that accounts for some huge proportion of risk.
Yeah, I mean, it's an argument that hasn't really been had, I don't think, enough about where these large resources of funding should go. Genetic drift is a really powerful and amazing thing, and these natural experiments, well, laboratories, I don't know what you want to call them, experiments of nature have been going around in India and all over the place, and it's to try to take advantage of that and learn from that. There'll be all sorts of loss of function variants that have drifted up, and every which kind of variant that will have drifted up to a frequency where you can actually do something about it and learn from it. Sometimes, observing one person isn't really enough, but once you've got 10 or 20 of them in your hands, you can see what's going on. So I agree. I think that would be very interesting. I would go for multiple. We can argue about how you do it, but each one is going to tell you something new. So it would be cool.
Yeah, and sometimes I'm thinking of in East London Genes and Health, and we had Paul Noy from Alnylam. In that case, there was literally one person that helped them crack the case on the HA01 gene. It was an ultra rare double knockout of this gene. They found a single person. They got consent to collect some new samples and actually validate a hypothesis of what was going on biochemically. And sometimes it is literally a single person with something that you'd need to sequence 10 million people somewhere else to have any hope of finding it. But if you sequence 100,000 in a specific population, then you find it. It's fascinating. I think we have to think about how to scale it up somehow. Is there a conference that people like you get together and all the isolated community genetics population, or is that ASHG?
Yeah, not at the minute. There was a series of conferences about isolated communities that ran in Italy for a number of years, but that's in abeyance at the minute. So yeah, we just go to the European and the American Society meetings pretty much.
Yeah, makes sense. I am kind of interested in the whole newborn screening movement, and there's a conference that's popped up, Icons, that is all about newborn screening. And this makes me think that it'd be interesting to, even if it was 1 day or 2 days in a easy to get to city, you could probably have some amazing people together. It sounds like it has been organized and is waxing and waning.
Yeah, no, I just heard about the Icons conference the other day as well. It's interesting. The world's moving forward.
Yeah, it is. Jim, thank you. This is amazing. I learned so much. You packed a lot into this 45 minutes that we had. So thank you. Appreciate you taking the time to be with us.
My pleasure. Thank you very much for the invite.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.